Hello, thank you for attending our webinar today. My name is Derek Schneweis. I'm a product marketing manager at GENEWIZ and Brooks Life Sciences. Today, we will discuss high-throughput antibody discovery at single-cell resolution. Here's the agenda for my talk today. We'll review challenges with current low-throughput antibody development methods, discuss next-generation antibody discovery using NGS approaches, review best methods for antibody lead optimization, and finally, we'll explore cell line development solutions for reducing your in-house processes and utilizing GENEWIZ services. So traditional antibody discovery workflows are ultimately low throughput. Here's a list of those traditional workflows, and you can see that no matter what the starting material is, hybridoma cells or recloning from B cells, limitation and reduction in throughput occurs early upstream at the stage where you isolate and screen individual cells. And you can see those steps here. Looking at the overall antibody therapeutics workflow, you can see ranging from discovery to preclinical to clinical, low throughput upstream really reduces your ability to discover antibodies fast, and it increases the complexity and upstream timeline for therapeutic delivery. If you look specifically at number 3 here for lead generation, you can apply single-cell immuno-profiling, which is an NGS method that'll expand and accelerate lead generation. Single B-cell sequencing is a high-throughput method to increase discovery early in the upstream process. Here you can see a generalized process, where you've challenged your species of interest with an antigen, collected PBMCs, performed library prep to capture the heavy and light chain sequences from B cells, and then sequenced using NGS. This allows rapid identification of a large panel of candidate sequences and removes the upstream bottleneck. It also allows you to discover more candidates, which we'll take a look at in future slides, and it enables collection of frequency data for heavy and light chain lineages. Starting with whole blood, you can further streamline your discovery. So here's an example of PBMC isolation at Brooks Life Sciences, where you're starting with whole blood. We'll also do the fractionation, the isolation, and the cryopreservation. You then move that sample directly to GENEWIZ for your single-cell immunoprofiling through the process that I described before. So this allows you to do high throughput capture and sequencing of single B cells directly from sampled blood. Here's an example of single-cell sequencing for rapid discovery of diverse antigen-reactive antibodies. This is a case study. You'll see on the right how the process is similar to what I've described before. B cells are sequenced as single individual cells. The bar-coded cDNA is used for library construction with retention of linked heavy and light chain regions. That enables deep sequencing of the VDJ regions and continual tracking of those two linked heavy and light chains. This approach provides comprehensive characterization of the entire B cell or T cell repertoire, and it's achieved through microfluidics. The heavy and light chain pairing provides prevalence data. The animals used in this dataset were not immunized with a target antigen, so we're looking at sequence data for naive antibody profiles. The heat maps on the right show the percentage of lineages with particular heavy and light chain pairings, and you can see some of the germline gene segments are more frequent in the dataset, and they're represented by darker red dots. Those higher than expected pairings might be due to immune responses to common antigens. So this type of frequency data is not common in traditional antibody discovery methods, and it might prove beneficial to your antibody discovery project. In this case study, scientists also looked at recovery of control lineages. They are ultimately able to identify more candidates when compared to traditional hybridoma screening. In this experiment, rats were immunized with chicken OVA. You can see on the right, using single-cell sequencing, scientists were able to recover 81% of anti-OVA lineages identified from the control hybridoma experiment. They were also able to identify an additional 710 candidates. Using SPR, they showed binding affinity for both the common lineages across the control and also the new candidates discovered. So you can see single-cell sequencing provides not only rapid discovery, but discovery of more candidates as well. Okay, so let's take a step back and look at that overall workflow. Here's it represented again, where we're talking about discovery to preclinical to clinical. What I want to do next is focus in on lead optimization. Lead optimization has many options. Here I've taken our next step into optimization using phage display, which you can see on the right. So you can pull your discovered antibody from single-cell NGS discovery, as I described before, and introduce it into a method like this. Once you have your clone, you can isolate and perform sequence optimization by site-directed mutagenesis or optimized by variant synthesis, which you can see in green. The next slide has some examples of synthetic DNA libraries for lead optimization. Here are some examples of DNA libraries that you can use for lead optimization. You have libraries like combinatorial libraries to generate mutants in multiple combinations. Tri-mer controlled libraries offer great control of amino acid distribution. They also eliminate the introduction of unwanted codons and maximize downstream efficiency. You can also use alanine scanning libraries or truncation libraries to identify the critical amino acids that are important for the CDRs. And then you have random libraries that create random variants for a selected region and are useful to enhance your lead stability and affinity maturation. Once you get through lead optimization, your next big step is candidate testing. So candidate testing success depends on quality and control of the cell line, right? So for your pilot or first lot manufacturing material, that's what you're going to actually use often when testing for efficacy and safety in preclinical. So when you think about that, your antibody cell line development has a bunch of different steps, right? That range from RUO, GLP to animal free. It's really important that you get that pattern correct and you approach your preclinical and then eventually clinical with the correct types of material and the correct definitions around that material. That's not an easy process, right? So GENEWIZ can help at multiple steps in your development process. Here I've depicted in an image different steps that you will likely go through as you develop your antibody or drug. All of the steps that are highlighted in blue are places where GENEWIZ can help you move your project on to the next phase. So you can see it ranges from gene synthesis and cloning to R&D and GLP levels of animal-free plasmid preps, Sanger verifications, even down to high-throughput cell line screening. So it's important to see that there's a bunch of steps as you get to that IND filing. So remember, it's important to think about how you plan to do that or if you need help as you work through your antibody development. Okay, so here's the whole workflow. We focused a lot in the upstream areas with discovery where we talked about next generation sequencing at the single cell level. That technology provides solutions really for high throughput, high resolution immunoprofiling, early antibody development. It provides a lot of options for screening and also gives you heavy and light chain pairing so you can track your heavy and light chain as you discover different antibody segments for your target antigen. You can see here there are many other steps that we covered throughout our talk. It's important to realize that each step has its own challenges and GENEWIZ is here to help. So you can see I've talked mostly about processes, but we also have ability to store and manage your sample as well through Brooks Life Sciences. So you can manage your sample at Brooks and send it to GENEWIZ for different types of genomics approaches. So keep that in mind as you progress your research. One other thing to keep in mind is that single-cell sequencing is not the only method for increasing throughput. High-throughput drug development is a rapidly evolving field. Transcriptomic profiling is a phenotyping tool that can further streamline your development process. If you're interested in increasing your drug discovery throughput, keep an eye out for our early access program at GENEWIZ. We've developed a high-throughput gene expression screening method for rapid cell-to-data. It's low cost and offers unbiased coverage, something that microarray cannot achieve, and it's useful for up-down selection of drug candidates. It is a screening method with a fast turnaround time for quick target discovery and candidate selections. We're very excited to show it to you. Thanks for joining me today for this webinar. And as always, seek more, learn more. Here's an image of our website if you want to find where we discuss single-cell antibody sequencing for early antibody discovery. I've also put a link here and a QR code if you prefer to use your phone and scan that. Both will take you to the website. Please feel free to reach out if you have any questions as you move forward in your research. And thanks again.